Okay. There you are. Okay, folks, you probably saw me handing out a lot of exams here. I hope that's not reflective of attendance, because if it is, then we may be stopping the videos. I don't want to do that. So I understand maybe you didn't pick them up after class yesterday, but as I said earlier in the term, if I see that the videos are causing attendance to drop, I will stop doing the videos, and I don't want to do that. So I expect you to be here for the most part, okay? All right. I forgot to mention yesterday that I was on the Nutrition Now radio show with one of the students in the class here, and I meant to mention it to everybody so you could listen to KBDR and hear it. Did anybody hear it? Oh, good. Okay, you won a gold star A right there. I'm told that the podcast of that will be available soon. And how do they find the podcast?
If you go to Oregon State's website, you can search KBDR, and the radio station link will come up, and you can listen now again.
Excellent. Okay. And if you can't find it, let me know, and I'll point you there. And a question on the final exam will be on the from the podcast, right? That's a joke. Okay. All right. All right. So I'm going to finish talking about energy today, and then I'm going to talk something about structure of sugars, okay? Structure of carbohydrates, which is a little bit of the subject that we talked about on the show last night. The show concerned sugars and artificial sweeteners. So that was really what we talked about on the show last night. Okay. Yesterday I introduced the topic of energy, and I gave you some examples for how that equation tells us something about the direction of a reaction. And at the very end, I made a comparison between the Henderson-Hasselbach equation and this equation. I want to reiterate that because they're really very similar. Okay? The Henderson-Hasselbach equation says pH equals pKa plus log of salt over acid, which means that the pH, which is a variable, depends upon a constant, which is the pKa, plus another variable term, the log of salt over acid. So as the salt and acid change, so does the pH change, right? This says the same thing. The delta G for a reaction, which is a variable, is equal to a constant plus RT times the natural log of these variables right here. So as products and reactants of a reaction change, so too will the delta G value change. And as I mentioned in class yesterday, the only thing cells have control over are these two guys right here. They've got to work within these parameters in order to make reactions go. And sometimes cells want a pathway to go forwards, and sometimes cells want a pathway to go backwards. We'll actually see that as it relates to sugar metabolism, and understanding that forward-backwards nature is important. If you look on the schedule page, you'll see some practice Gibbs free energy problems that I assigned, and I want you to make sure you know how to work through those, okay? So you probably will see something like that on the next exam, which, by the way, is a week from tomorrow. Okay. Now, I like this diagram. This diagram, well, the reason we talk about energy in the class is cells have to get energy to do what they do. They have to have energy to communicate with each other. They have to have energy to make proteins. They have to have energy to make DNA. They have to have energy if they're a muscle cell to contract. All kinds of things that cells have to do, and all of them require energy. Well, as we all know, the ultimate source of energy on the face of the earth is the sun, which beams energy to us through the fusion of hydrogen to make helium. Okay? So plants capture all of that energy on Earth in the form of photosynthesis, and ultimately everything we have on Earth happens as a result of this process right here. Photosynthesis is not just an energy capture, but it's an efficient energy capture system. And what it's doing is, if it simply captured energy, that really wouldn't be real useful unless it captured it in some form it could do something with. And that's exactly what photosynthesis does. Photosynthesis captures energy and stores it chemically, or biochemically. Okay? The biochemical storage is in the form of glucose. So plants are taking out of the atmosphere carbon dioxide and water and using those two together to make glucose. They also, in the process, release oxygen. The reason we have as much oxygen in the atmosphere as we do is because plants have been working on putting that oxygen out there for billions of years. The early Earth had very, very, very little oxygen. Okay? Today, oxygen is about 20% of our atmosphere, and that's, while that might sound like a low percentage, it's a tremendous amount of oxygen, and it's happened because of photosynthesis. So that's a very, very important consideration. What do? Well, we take that glucose, we break down that glucose, we regenerate carbon dioxide and water, but we're capturing the energy now of the glucose in the form of ATP. That's what we're doing. Okay? Now, we use other energy sources besides glucose, but from a simple perspective, that's what we're doing. We're taking energy from the oxidation of glucose and making ATP, and that ATP is important for us for all the things that we do. ATP is what I like to refer to as the cell's energy currency. Just like a dollar is the currency in this country, I can take a dollar into virtually any store and buy something with it, okay? Unless it costs more than $1, of course, in which case it might take $2. So too is ATP essentially universal within the cell and between cells. It's a currency. It's very, very commonly used. Well, the energy of ATP is, of course, used to make proteins. It's used to make DNA. It's used to make fat. And all of these, of course, together are used by cells as part of their normal metabolic process. So this energy flow is really, really important for us to understand. Another way of looking at this, this introduces a couple terms to us, is in view of what are called catabolism and anabolism. Okay? Catabolism refers to pathways that are catabolic, not diabolic, catabolic. Diabolic is something else. Okay? Catabolic pathways are degradative in nature. They break bigger things down into smaller things. And catabolic processes are generally oxidative, meaning oxidation is involved. Another characteristic of catabolic processes are that they are energy releasing, because oxidative reactions are pretty much energy releasing. Okay? So if we start at the top and we move down, we're basically going in a catabolic direction, from large things like fats, polysaccharides, and proteins that we're breaking down into smaller molecules. We're going downwards. We're doing catabolic things. If we are synthesizing, that is, if we're making these compounds, we're doing what's called anabolism, or anabolic processes. Okay? So anabolic processes move upwards. They start with simple, small things. They use energy to put those simple, small things together into bigger things, and then ultimately into the very biggest things of all. Anabolic processes are generally the opposite of catabolic. That is, instead of being oxidative, they are in general reductive. Okay? They're in general reductive, and they are energy requiring. Okay, so catabolic and anabolic processes, very important to understand. Now, I like to show this figure because it kind of gives us an idea about what we call states of oxidation. Different states of oxidation. Okay, we talk about oxidation. I'm going to show you some examples of oxidation in a minute, but this illustrates it pretty well. If we look at these various oxidative states of carbon, they go from the most reduced on the left to the most oxidized on the right. And that should have a C double bonded to oxygens. The C disappeared somehow. Okay? This is the most reduced. Okay? Carbon with no oxygens on it. Here's a carbon with a hydroxyl on it. That's slightly more oxidized. A carbon with a double bonded oxygen, slightly more oxidized. A carbon with a double bonded oxygen and an OH, more oxidized, and a carbon with two oxygens, completely oxidized. Okay? As we go from this compound to this compound to this compound to this compound to this compound, each time we take one step to the right, we release more energy, because each of these is an oxidative step. Each of these is an oxidative step. Okay. When we talk about oxidation, it's important to recognize that wherever there's an oxidation, there is also always a reduction. It's like for every reaction, there's an equal and opposite. For every action, there's an equal and opposite reaction. For every oxidation, there's an equal and opposite reduction. The reason that happens is because oxidation involves the loss of electrons. Well, those electrons don't get destroyed. They have to go somewhere. And when they go somewhere, they join with something else and reduce it. Okay? This shows the oxidation of ethanol. Ethanol can be oxidized. When it is oxidized, it creates acetaldehyde. And if you want to know the reason you get a hangover, there's a good reason right there, acetaldehyde. Okay? This shows simply that the electrons and the protons are lost. It's actually the loss of electrons that's the oxidation. But fortunately, biologically, we don't have this happen. We don't have free electrons that are released from this oxidation process, because if we did, those electrons would combine with compounds randomly, number one. And number two, those random acceptance of electrons would create extraordinarily reactive molecules. We can't have that in a cell. Cells are control freaks. They can't have this very, very reactive species being created. Okay. Well, what cells have is they, instead of releasing electrons freely, they use what are called electron carriers. So the electrons are given to molecules that have special characteristics that allow them to accept electrons. One of these compounds is known as NAD, nicotine adenine dinucleotide. You're more likely to call it NAD, just like I am. Okay? When NAD has not yet accepted electrons, it's called NAD. After it has accepted 2 electrons and a proton, it is called NADH. The only difference... Yes. So the molecule that has not accepted electrons is called NAD. After it has accepted 2 electrons and 1 proton, it's called NADH. Now let's think about this. NAD is positively charged. NADH is not charged. Why is that? Well, if we had something that was uncharged, if we had something that was positively charged, and we added 2 electrons to it, well, what does charge be? -1, right? +1 minus 2 is equal to -1. And we add a proton to it, what's the charge? 0. Okay? So that's why it has a charge of 0. All right. So if we took this bottom portion and we connected it over here, we would have NADH. This is just simply showing us where the electrons are going on. You won't have to draw this structure. That's not what this is about. But you should know that NAD is the more oxidized form because it's lacking the electrons. And NADH is the more reduced form because it has accepted those electrons. All right? Now, as we look at this, all right, here's another molecule I should point out, okay? Here's another molecule that accepts electrons, FAD. FAD, when it is in the oxidized form, is called FAD. FAD, however, accepts 2 electrons and 2 protons. It becomes FADH2, and notice that its charge does not change. You start with 0, you add 2 minuses, you add 2 pluses, you still end up with 0. Okay? In the cell, when I oxidize ethanol, okay, I said I don't lose the electrons. Instead, they go to an electron carrier. So the reaction that I would write for the oxidation of ethanol would go as follows. Ethanol plus NAD goes to acetaldehyde plus NADH. Now let's think about what I just told you. Ethanol plus NAD. Ethanol is the reduced form because it has the electrons. NAD is the oxidized form because it doesn't yet have the electrons. Acetaldehyde, however, has lost 2 electrons. It's the oxidized form. NADH has accepted electrons. It's the reduced form. So just as I said before, wherever we have an oxidation, there must be a reduction. As ethanol gets oxidized, NAD gets reduced. They always pair in that way. One molecule gets oxidized, the other molecule gets reduced, because that's what's happening in reduction, is those electrons are going to something else. Okay. Questions about that? Okay. There's a big bunch of structures. Question? Okay. All right. Well, the other thing relevant for us with respect to energy are high-energy phosphates. Okay? And by the way, when we talk about electrons with NAD and so forth, it turns out that NADH has energy because it has those electrons. We'll see later how that energy is used by the cell. Okay? Ultimately, the energy that's in NADH can be used to make ATP. We'll see how that happens later. Okay? But right now, I want to turn our attention to talking about ATP. ATP, as I said, is the energy currency of the cell. The reason ATP has such high energy is because right here, these phosphates are all negatively charged, and they all repel each other. They all want to go flying away, and they're held in place by covalent bonds. It's kind of like when you're a little kid and you get in trouble with your sibling, which I did a lot, and your mother made you go and sit in the room with them and behave yourselves, right? And you really didn't want to be in that room with your sibling behaving yourself. Well, here's 2 siblings that don't want to get along, and they're held together by a covalent bond. If you were like me, you wanted to get out of that room with your sibling as quickly as you could, and so too do these phosphates want to get out as well. This simply shows what happens. All right? To release energy from ATP, we hydrolyze it, meaning we add water. When we add water to it, a phosphate gets clipped off. And yes, that phosphate goes flying away. Similarly, we can release energy from ADP. It's not done nearly as commonly, but that can also happen. And it also releases energy. Okay. Now, this table I find really interesting. Okay? Look at the energy of ATP. Here's the delta G 0 prime. That's the energy of hydrolysis of ATP. That's how much energy ATP would give up under standard conditions. And what were standard conditions again? Temperature, pressure, and 1 molar everything, right? So if I have equal concentrations of ATP and ADP, then this is how much energy is released there. Okay? Minus 30.5 kilojoules per mole. That's actually a very negative number. It means that if I have these guys present in equal concentrations, that's what the delta G value would be. Okay? A negative delta G value tells us it's going forward, and a large negative delta G value tells us it's going strongly forwards. Okay? Well, look at this guy here. ATP's got a lot of energy, but there are things in the cell that have higher energy than ATP. How do we make them? How do we make them? Okay? Well, cell's got some tricks up its sleeve, and some of those tricks are kind of cool. They illustrate for us how the principle of ATP, how the principle of concentration is used. Okay? I'm not going to go through those right now. There is at least one of them that's illustrated for you in the Gibbs problems that I have for you on the schedule page. Look at those, and if you have questions, let me know. I'll be happy to work through those with you and make you understand that. Okay? Well, this figure shows us now how ATP fits in this process. Okay? Catabolic processes, okay, like burning of glucose, gives us ATP. ATP is used to drive anabolic processes to make things that cells need. Okay, so it's just showing us in a figure something similar to what we've already seen before. And I'll save that for later. Okay. That's energy. Questions about that?
Can you repeat that last bit where you said catabolic processes
Yep.
give ATP?
Sure. Okay. And blah, blah, blah. I like that part. That's my favorite part. Okay. So the blah, blah, blah is that this is what's happening in our bodies. We are taking food sources, like in this case, glucose. Glucose is also an energy storage form. We oxidize glucose. That's where this oxygen comes in. And when we oxidize glucose, our cells use the energy of that oxidation to make ATP. They convert ADP into ATP. So the energy of that oxidation creates ATP. ATP now can be used for many things inside the cell. Particularly in this case, it can be used for anabolic processes, that is the making of things that cells need. So ATP energy can be used to make DNA, it can be used to make RNA, it can be used indirectly to make protein. Remember, we use GTP, but ATP can be readily made into GTP, so we essentially think of it as the same thing. Okay? As a consequence of this, all right, we can make things that we need. We can make the things that we need. ATP, I like to also think of as the gasoline of the cell. Okay? Gasoline takes you places. Gasoline can be used for heat. Gasoline can be used for generating electricity. Gasoline can be used for a wide variety of things. And ATP functions in a very similar fashion because ATP is providing the energy needed to make things, just like gasoline is providing the energy needed to move things or heat things. Yeah?
So you're talking about how translation requires GTP. Is that the only process in the body that uses GTP?
No, that's not the only place in the body where GTP is used because GTP is necessary to make RNA. Right? A, G, C, and U, we're using GTP every fourth nucleotide, right? Okay, so yes, GTP is used in other places. There are some other metabolic processes that use it as well. We'll see in the citric acid cycle that there's actually a reaction in the citric acid cycle that makes GTP. So there are other places it can be used as well. Okay? All right. Well, let's turn our attention to talking about carbohydrates. This, I find, for most students is pretty much review from organic chemistry. So hopefully you can sit back and relax and listen to a bunch of terms that probably you've heard before. Now, before we do that, maybe we should sing a song. I've got a song for energy. You want to do that? Let's do a song. This one's to the tune, we've used this tune before. It's to the tune of Danny Boy. It's called O Delta G. O delta G, the change in Gibbs free energy, can tell us if a process will advance. 'Cause if the value's less than naught, it translates that reverse reactions haven't got a chance. But when the sign is plus, it is the opposite. And then the backwards happens all the time. A factor is the standard Gibbs free energy. So don't forget about the delta G naught prime. Okay. All right, carbohydrates. Carbohydrates are pretty hot topics. They certainly were last night on Nutrition Now, and they certainly are in our understanding about how our bodies function. Okay? Very, very important things, and we're learning a tremendous amount about carbohydrates and their effects on our physiology, how we do our day-to-day life, and how it involves and affects processes, very important processes like diabetes that occur within us. The name carbohydrate tells us, first of all, what we're working with. Carbo referring to carbon, hydrate referring to water. Carbohydrate. Okay? What does that mean? Well, the simplest carbohydrates are shown right here. Glyceraldehyde and dihydroxyacetone. All right? If we look at the structures of those, we see that they each have the structures C3, H6O3. If we look at the next most complicated ones, we see they have the structure C4, H8O4. If we look at the next most complicated ones, they have the structure C5, H10O5. And if we look at glucose and its related compounds, they have the structure C6, H12O6. There's a pattern there. There's twice as many hydrogens as there are oxygens, and there's an equal number of oxygens as there are carbons, meaning I could write this as C with a little X and an H2O with an X as well. Carbohydrate, carbon hydrate. That's what that is. Okay, so that's where the term comes from. Okay, as I said, these are the simplest of the carbohydrates here that we find in the body. And these compounds are what we refer to as trioses. Whenever we see in biochemistry a name ending in OSE, it always tells us that that molecule is a sugar. So triose tells us, tri meaning 3, that this is a sugar that has 3 carbons in it. And that's exactly what these guys have. 1, 2, 3. Okay? Now, so far in the class, I haven't made you memorize any structures, but we're going to memorize a few structures in the carbohydrates. And the reason I make you memorize a few structures is because they're structures you will need to know in other classes. Okay? There's only a few. I will assure you of that. But there are a few that you need to know, you need to memorize, because I guarantee you'll see them again. Okay? Well, one of the things that we see about carbohydrates is that generally they have at least one carbon that has 4 different things attached to it. We've seen this before in amino acids, and now we see it appearing in the carbohydrates. Okay? The very simplest guys here, all right, here's glyceraldehyde. Glyceraldehyde has one carbon that has 4 different things attached to it. Okay? 4 different things attached to it. That's what it looks like. Okay? And there it is. These are shown in perspective here. Deglyceraldehyde is drawn here. L-glyceraldehyde is drawn down here. And biochemists, by the way, are very lazy. Biochemists use D and L designations. We don't use R and S. And you'll see D and L is very easy to learn. Okay? For the moment, you're going to look at this, you're going to say, oh, well, the L, it's got the hydroxyl on the left. And that's right. Okay? It's a little bit more complicated than that, but that's basically what it is. The hydroxyl's on the left. All right? Notice that dihydroxyacetone doesn't have 2 different forms, and that's because there's no 2 carbons in, there's no carbon in there that has 4 different things attached. That's because the middle carbon has a double bond to the oxygen there. And that's that. Okay. Here is the structure of 2 molecules that you need to know the structure of. Glucose and fructose. Okay? This shows the structure on the top of D-glucose and D-fructose. Glucose is, for example, an example of what we would call a hexose, meaning it has hex meaning 6 carbons. If we count the carbons in fructose, we'll see it also is a hexose. It also has 6 carbons. But we see there is a chemical difference between them. The primary chemical difference between them is that glucose has an aldehyde group and fructose has a ketone group. And that should be a double bond oxygen. I always don't catch those before I get in here. Double bond oxygen, okay? Not double bond OH. All right. I'll change that. If we look at that, okay, that's an aldehyde, that's a ketone. We call glucose as a consequence an aldose. We call fructose a ketose. Okay? So then we can say, okay, so if we were to describe glucose, we would say, well, it's an aldohexose. Or is it a hexoaldose? And the answer is it doesn't matter. As long as you know the categories it fits into. So this guy's an aldehyde. This guy is a hexose. Well, this is D. Okay? These guys are both in the D configuration. And by the way, for almost all sugars we find in the body, they're in the D configuration. Almost all. Not all, but pretty much the vast majority of sugars we find in the body are in the D configuration. Okay? On the bottom you can see L-glucose. And you look and sure enough, there's that hydroxyl on the left side. But we notice something else. First of all, glucose and fructose have multiple carbons that have 4 different things attached to them. Multiple carbons. So the left or right nature of that OH really matters. When I was in biochemistry the first time, the way I memorized the structure of glucose was right, left, right, right. Right, left, right, right. Okay? If we look at fructose, it turns out to be the same, except for this double bonded oxygen here. Left, right, right. The bottom 3 are the same. So if you know the structure of glucose, you essentially already know the structure of fructose. Right? Well, look at L-glucose. L-glucose turns out to be, and this is a very important point, the mirror image of D-glucose. It doesn't just differ right here. We have to flip every one. So where this guy's on the right, this one's on the left. Where this one's on the right, this is on the left. Where this is on the left, this is on the right. The difference between a D sugar and an L sugar is that they are mirror images of each other. D-glucose is a mirror image of L-glucose. Okay? Now, that is an important distinction that, surprisingly enough, even majors in biochemistry don't remember sometimes. So you guys will do me very proud if you remember that. Okay? Very proud if you remember that. D versus L. All right? Okay. We give a name to to sugars specifically that are mirror images of each other. They're called enantiomers. E-N-A-N-T-I-O-M-E-R-S. Enantiomers. Okay? I hear a sigh. Yes. Okay. Molecules that have the same structure, but that differ in their stereoisomeric configuration are called stereoisomers, right? So mirror images, for example, are stereoisomers of each other. Well, what about things that are not mirror images of each other? These guys are all mirror images. Here's D-erythrose. Here's L-erythrose. We can see these guys are mirror images. And here's D-threose, and here's L-threose, and these guys are mirror images of each other. Okay? Well, what if we were to compare D-erythrose to D-threose? They're not mirror images of each other. This guy's got them both on the right. This has one on the left and one on the right. Right? So they're not mirror images. Molecules like this we call diastereomers. D-I-A-S-T-E-R-E-O-M-E-R-S. Diastereomers. So stereoisomers that are mirror images are enantiomers. Stereoisomers that are not mirror images are known as diastereomers. Mouthful of names. Now, they have to have the same number of carbons, obviously. They have to both be aldoses, as in this case, or they would have to both be ketoses. They have to be the same type of sugar. But if they have those characteristics, they're either enantiomers or they are diastereomers. Oh, boy. Now, this figure is really not bad. It shows us what happens every time we add a carbon to a simple sugar. Here's a simple sugar that's got 3 carbons. Here's one that's got 4 carbons. These have 5 carbons. These have 6 carbons. And notice that every time we add a carbon, we add 2 more structural possibilities because each one can have left and right, right? So we double, double, double. One of the good things is we don't get sugars too big. If we did, we'd have so many possibilities, we'd probably run out of names. And no, you're not going to memorize these. All right? I will tell you the ones you're going to memorize. This guy, ribose. Okay? Ribose is essential to know. First of all, ribose is a pentose. It has 5 carbons. It is an aldose, meaning it has an aldehyde group. Most importantly, ribose is found in ribonucleotides. That's where the ribo part comes from. Ribonucleotides. Now, structurally, this guy is very trivial to memorize the structure of. 1, 2, 3, 4, 5 carbons, aldehyde, all the OHs on the right side. It's pretty easy. Okay? When we get to the 6 carbon sugars, we've got glucose. Right, left, right, right. Okay? I think you also should know the structure of galactose. Galactose, we shall see, has some important chemistry for us. And galactose is found in lactose, the sugar most commonly found in milk. All right? Galactose is related to glucose. It only differs right here with this carbon, carbon number 4. Instead of right, left, right, right, it's right, left, left, right. So far you have to know the structures of ribose, glucose, galactose, and I'm going to show you fructose in just a second. Okay. Oh, it's actually back here. Well, actually, back up. Sorry. Fructose Okay, fructose was here. That should be double bond oxygen, not double bond OH. But that's fructose. And as I said, fructose has the same structure as glucose, just a ketose instead of an aldose. Okay, so those... Oh, that's okay. It woke everybody up, including me. So those are the structures of carbohydrates that you need to know. All right? Now, what you may remember from your organic chemistry is that sugars don't exist solely in a linear form. They can cyclize. And this shows the cyclization. All right? Here's glucose in a what we call a straight chain form. This form is called the Fischer form, F-I-S-C-H-E-R. The Fischer form can cyclize. Carbon number 5's oxygen can attack that guy. And when it does, it makes 2 different cyclic forms. 2 different cyclic forms. All right? Now, what these 2 different cyclic forms, the reason we have 2 different cyclic forms is we started with a carbon that was not asymmetric, and we've just made it asymmetric. All right? This carbon number 1 now has 4 different things attached to it, and consequently, those things can be arranged in 2 different ways in 3-dimensional space. Okay? We refer to these 2 different forms in the cyclic form as the alpha and the beta form. The alpha will always have the hydroxyl down. The beta will always have the hydroxyl up. Now notice that we only have alpha and beta when we have the circular form. When it's linear, we don't have an asymmetric carbon. Okay? The circular form also has a name. It's known as the Hayworth form. Hayworth, H-A-W-O-R-T-H. The Hayworth form is a circular form of sugars. Most sugars can exist in both linear and in circular form. These 2 sugars are described as anomers. A-N-O-M-E-R-S. Okay? They're anomers, meaning that they differ only in the configuration of this carbon right here, which is called the anomeric carbon. So an anomeric carbon is the newly asymmetric one that forms during cyclization. Okay? Sure. All right, so let's say this again. So we've got these two guys are called anomers. And anomers are sugars that differ only in the configuration of the newly asymmetric carbon. The newly asymmetric carbon is the one that had the aldehyde. We shall also see, when we look at ketoses, they can also do this, and they will form that with the keto group. All right? But anyway, these guys are anomers. This is the anomeric carbon. Anomers differ only in the configuration of the anomeric carbon, meaning that if I have alpha D-glucose and I have beta D-glucose, those two molecules will be anomers. If I compared alpha D-glucose to beta D-galactose, they're not anomers because it's not the same sugar.
Is it Anomer?
A-N-O-M-E-R, yes. Anomers. Okay. All right. By the way, your book calls this a fissure. I don't use that designation for this. Okay. I call this fissure. Okay. Over here. All right. Glucose. There we go. All right. Another thing I want to talk about is thinking about these cyclic structures that can exist. Okay? The cyclic structures that can exist, there's actually some bend that happens with the bonds. And those bonds can have different configurations of bending. Okay? So, for example, this illustrates for us a little bit better in three-dimensional space one of the ways that those bonds can bend. This guy's called the chair form. And the reason it's called the chair form is because this looks like the head of the chair where you sit in your butt, and then there's where you put your feet down here. Right? Looks kind of like a chaise lounge. Right? Well, this very same sugar can exist in another way, still in the alpha configuration, but in which the bonds bend a little bit, and this guy goes up. It's called the boat form because it resembles a boat. Okay? Oh, I thought I had a structure there. All right. If I had this same guy right here, and these guys pointed up like that, then it looks like a boat, a canoe or something, right? They're both glucose. They're both in the alpha configuration. It's just how the bonds bend with respect to each other. So boat and chair are two possible conformations that can exist for alpha D glucose. Now I'd like you to think about this. Let's imagine I bend this guy up, right? When I bend this guy up, this hydroxyl and this H, which is here, now starts encroaching on the space of where this CH2OH is. We think about steric hindrance. We've talked about steric hindrance in class. We know that atoms don't want to get too close to each other. And though these guys don't get overly close to each other, they're closer than they would like to be to each other. So when we look at glucose, alpha D glucose like this, what we see is that alpha D glucose prefers to be in the chair form, not the boat form. It will exist in the boat form, but only in a small percentage. Most of the molecules will be in the chair form. Okay. I'm sorry? What causes it to go from one to the other? It's just randomness. There's flexibility in those bonds that are there, and so that flexibility actually gives rise to that. But because there's randomness and because that this random structure is less stable than this one is, then the chair is preferred over the boat. Okay? Okay. Now, here's a dimer of glucose. What we see that happens with carbohydrates is they can be strung together. We can link one to another, to another, to another, to another. We can make polymers of these guys. And polymers turn out to be very useful for us because they allow us to store large numbers of sugars in a fairly compact place. Okay? Glycogen is a polymer of sugar, of glucose that's in our body. A polymer. It will have thousands of glucoses contained within it. Okay? So stringing sugars together turns out to be very, very useful. And what you see on the screen is the way that they are strung together. This was a free glucose. This was a free glucose. What we see is carbon number 1, 2, 3, 4 is linked to carbon number 1 through a bond like that. Okay? But what did I do with this in putting carbon number 1 with carbon number 4? I've actually combined the OH that was on that anomeric carbon. That turns out to affect the chemistry of that OH. It affects the chemistry of the OH. Yeah. Carbon number 1, 2, 3, 4. Okay, carbon number 4 is ended up being bonded to the hydroxyl of what was the hydroxyl of carbon number 1. That anomeric carbon no longer has an OH on it. It has an O bonded to something else. That changes its chemistry. It makes it much less reactive. Okay? Because what we have made is something called a glycoside. Glycoside. Glycosides are chemically stabler than are non-glycosides. Okay? They're less likely to oxidize, much less likely to oxidize. Okay? Than a non-glycoside. So if I had these both, I'm sorry? That's right. So if I look at this guy right here, this will readily oxidize. This will not. Okay? So that glycosidic bond is much stabler than an anomeric carbon there. Okay? Okay? Well, I've just talked about oxidizing. Let's think about the other way we can go. If we don't oxidize something, we reduce it. I'm just about done, so give me just a second. Okay? If we oxidize something, or we reduce it, look what happens, all right? Well, here's sorbos. Sorbos is a relatively simple sugar. It's related, well, I won't go through that, but it looks like this, okay? If we take that ketone and we reduce it to an alcohol, we create something called an alditol, or a sugar alcohol. Sugar alcohols turn out to have very low reactivity because OHs on there are very, very unreactive. Very unreactive. Okay? Sorbitol is actually used as an artificial sweetener because it fools your tongue into thinking it's sugar. But because it's not sugar, it's not metabolized like sugar, even though it tastes like it. Much less caloric content because you really don't break down sorbitol very well. It tends to pass through you. Okay? It's one of the reasons it's used as an artificial sweetener. Okay, let me stop there. I'll say more about that, and I'm sure I'll have more questions about artificial sweeteners tomorrow. Yes, sir.
Regrades about the exam.
Okay.
Will be taken up until a week from today or a week from next?
From when I hand the back yesterday. Hi.
Just kidding.
What's your name?
Hopper, Eric.
Hopper, Eric. Okay. Yep.
quickly, I wasn't sure back in OCHEM. It said that was a new bond when you have the 1,4 magocytic linkage.
Yep.
It said it was a dimer. Does that mean covalent, or is that just a name?
Dimer means two things.
Two things.
Two
So
things.
this is a thymine dimer. That was just them two dimers.
Two thymines, that's right. Okay.
So it said at the end, I know xylitol and cellulose.
Yep.
Xylitol is used in the strength of teeth, right?
Xylitol is used actually also for artificial sweetening purposes, and it's not broken down readily. That's the reason these things happen. Go ahead. Yeah.
Yeah,